Okay, hi everybody. I'm just going to say a couple of words by way of introducing Dr. Riepe, who is a postdoc in my lab, as you just heard. First of all, I just really want to acknowledge CFRI for having supported Celeste and more broadly supported the CF research in my lab, really since the very beginning. Back in the 1990s. I just wanted to start off by saying, as Devin pointed out, I've been working on CF biology for a long time, including our discoveries in the early 1990s on the processing of the protein. But I thought I would out myself. This slide here shows me in 1969 at the age of 16, doing sweat tests at the Boston Children's Hospital. That was my summer job for through the end of high school, through the middle of college. And that was really how I got introduced, not only to cystic fibrosis, but it was through this man, Dr. Harry Schwachman, who ran the cystic fibrosis clinic, who really was one of the people who was most credited for defining cystic fibrosis clinically, and happened to be my pediatrician for reasons which I'm happy to explain over a beer, but became a friend of the family and gave me this opportunity, not only to do this summer job, but really inspired me to become a scientist. And so I really want to acknowledge Harry Schwachman as well. So with that, I just will turn over to Celeste to tell you about the exciting work that she's doing.
Hi, everyone. I'm super excited to be talking to you guys about my fellowship project, which involves understanding how corrected CFTR-F508 delta is targeted for endolysosomal degradation. So I realize that many folks here do not live and breathe cell biology like I do. So I'm going to give a brief overview of some basic CFTR cell biology. So just to start, CFTR is a chloride ion channel. So it forms a little pore in the membrane, and then specifically in the plasma membrane of epithelial cells. And of course, epithelial cells are cells that line the lungs, pancreas, intestines, as well as all the tissues that we know are associated with CF. Now, the way CFTR is made in the cell is that it is transcribed into messenger RNA in the nucleus. So we have a genome which contains our DNA and has two copies of the CFTR gene, one from mom and one from dad. And this gets transcribed into CFTR messenger RNA. Now that CFTR messenger RNA travels to the endoplasmic reticulum, where it is then translated into protein. And as it's being translated, it undergoes this process called core glycosylation, which is to add sugars onto the CFTR ion channel to monitor its folding state. And then from there, CFTR travels to the Golgi, where the sugars are further modified through this process called complex glycosylation. And from the Golgi, it then travels to the plasma membrane, where it then functions as a chloride ion channel. Now, a key experimental method that we use in a cell biology lab is immunoblotting. So we're detecting CFTR protein with antibodies. So in this experiment, we resolve the CFTR by its size. And for typical wild type, which is the non-disease form of CFTR, we get 2 bands. And this is the case when we're using stuff in cell culture, not necessarily true in tissues. We get 2 bands. And these 2 bands correspond to the core form, which is the ER form, and the complex form, which is the Golgi and plasma membrane form. Now, these are known as band B and band C, which you might see in some other talks. So once you get to the plasma membrane, mature CFTR gets recycled through the endolysosomal system. And this happens when you have too much CFTR and also when you have damaged CFTR. And what happens is that endosomes bud off the plasma membrane, and then they fuse with this, what I think of like the stomach of the cell. This is the lysosome. It carries a lot of proteases. It's acidic. And then that degrades CFTR. Now, the most common disease variant, F508 delta, misfolds at the endoplasmic reticulum. And it does indeed get this chloroglycosylation put on it as it is folding. However, it is identified by the endoplasmic reticulum associated degradation system. This is the protein quality control system in the cell. And E3 ligases put on ubiquitin, which serves as an eat me signal. And this eat me signal directs the protein to be degraded by the proteasome into its component amino acids. And this was characterized originally in the Kopito lab in the early 1990s. Now, one thing we can note when we look at immunoblots of F508 delta compared with wild type is that we notice two things. One, that there's less of the protein overall, and we also don't see any in the complex form. And that's because there's two critical defects that happen when you have F508 delta. One is that it's going through this process of ERAD, so there's just less protein overall, and it's not escaping the ER. So the most effective CF therapies employ these molecules called correctors. And what happens with a corrector is that it actually blocks, or I wouldn't say blocks ERAD, it diverts CFTR from ERAD. And then it enables CFTR to exit the ER and go to the plasma membrane where it can function as a chloride ion channel. So high throughput screens have identified two corrector drugs, elexacaftor and tezacaftor, and when paired with a potentiator drug, this becomes ETI. Now, a lot of my project is going to have ET in it, but I also want to acknowledge the fact that we also have some new correctors out on the market. These are vanzacaftor, tezacaftor, and tezacaftor, which form a liftric. I have a hard time saying this word, I'm so sorry, which is also known as VTD. And so I will be showing data from both sets of correctors. Now, correctors help F508 delta achieve the correct shape. So what I'm showing you guys here is this amazing atomic level wild type structure. And this is with cryo-EM. Basically, this group out of Rockefeller University, Ju Chen's group, this amazing job looking at the CFTR molecule at the atomic level. And so here you can see the two transmembrane domains of CFTR. And we can also see the two cytosolic domains, NBD1 and NBD2. And so we can get this really nice resolution. We can see, quite nicely here, there's this nice little pore through CFTR. And then when we look, or when the Chen group looks at the structure of F508 delta, they noticed that the area where F508 delta isn't particularly well modeled. There's a whole detergent cage around it. It's really hard to see. And that's because the protein isn't forming this nice 3D structure. And then when the Chen group added in ETI, they saw that it now formed a structure that is completely like wild type. So that's why they're called correctors, right? They help it correct, make F508 delta into the correct shape. So another thing that the Chen group saw is that ETI binds at a location in the CFTR protein that is way far away from F508 delta. And one of the really fundamental things about correctors is that they can work throughout the protein. And now we are at a point where 303 variants are approved for corrector therapies, which is just remarkable. If you look at disease variants of CFTR, so this is an image from a study from the Schlebach group at Purdue. This is looking at 129 different disease variants. You can see those disease variants are throughout the protein. And then when they looked and tested, okay, are these corrector responsive or are they corrector resistant? They found that the correctable variants are scattered throughout the protein. And then when they looked at resistant variants, they also found that they were scattered throughout the protein. And so there doesn't seem to be a great reason or some sort of system of logic for why certain variants respond and why others don't. And the truth of the matter is 17% of variants do not respond to correctors. And what I mean by respond to correctors is that they have less than 10% wild type function when we add in ETI. So this gets to the central point of my research, which is that about 15 to 20% of people with CF in the US do not benefit from corrector therapies. And so the questions that I'm interested in are how can we restore CFTR function in people with CF who cannot take the drug? And also, can we enhance corrector activity in people with CF who qualify yet don't respond particularly well? And so the way I go about this is I conduct unbiased screens for genes that could serve as drug targets for increasing corrector efficacy. And so I use F508 delta as a model for people who qualify yet don't respond well. And then I also look at other mutations throughout the protein. I look at I507del because it's really interesting. It's next door to F508 delta, yet doesn't respond. I also am looking at N1303K because it's in NBD2. It's also the second most common folding defect in the US. And I'm also looking at L227R, W1098R, and W57G, largely because they represent different protein domains in CFTR. So the strategy that I'm using for this is I inhibit or activate 20,000 genes in the human genome with CRISPR-Cas9, and then I have an assay to quantify the amount of plasma membrane expression. And then once we've come through and we've looked to see that, okay, yes, indeed, we can restore plasma membrane expression, we then plan on validating hits in airway epithelia with a collaboration with the MIA and Porteous labs. And of course, once we go through and we can validate this genetically, we hope to be able to send our data to people who work in chemical biology, who can use AI to predict drugs that can target these hits, and also build chemical libraries that we can test on CFTR function. So what types of hits do we expect to get in this type of study? So one is that there could be enzymes that break down correctors, and hyperactive versions of these enzymes could be interfering with the response. Another thing that we could find out is that there's quality control mechanisms that degrade corrected CFTR. Another thing is that there might be trafficking machinery that blocks CFTR export out of the ER. And if we interfere with that, we might be able to send more of the CFTR to the cell surface. So the way this works, or the way my assay works, is that I have a surface display reporter that can quantify the amount of CFTR that traffics to the cell surface. Now, the way this works is I have an N-terminal tag with a green fluorescent protein, and this enables me to quantify total CFTR as well as CFTR turnover. And then I also have an anti-HA antibody assay where I've engineered the HA epitope into CFTR, and now I can bind antibodies to the cell surface and use this as a quantitative readout. And I can also do some things to measure the amount that's trafficked by measuring the ratio of HA to M neon green. So surface display assay enables the quantification of CFTR in response to correctors. So as you recall, there are two fundamental defects when we were looking at F508 delta. There's no exit from the ER, and then the protein gets degraded through ERAD. And of course, when we add in the correctors, we then get ER exit, and then there's no ERAD. So with no correctors, F508 delta gets degraded and does not traffic to the cell surface. So what happens in our assay is the cells glow dimly green, and there's no red signal. And then again, because it's just not making it out of the ER. With correctors, F508 delta traffics to the surface where it binds to the HA antibodies. And now our cells glow green and red. Sorry. We wanted to work to validate that the system was actually working. And this is some real data that was collected by my amazing soon-to-be undergrad, Felicia, who is here up in the front. So what happened is we add some correctors to our F508 delta cells, and we saw that there was a threefold change in CFTR. So right off the bat, we're getting way more protein in the cell. We're also seeing this huge increase in the cell amount of CFTR that traffics to the cell surface. And then we can also see nicely that there's a big change in the ratio of HA to M neon green. So that's great. So now I want to talk about the other part of this reporter, which is this M neon green, which enables us to quantify CFTR turnover. So the way this works is that we quantify CFTR degradation in the cell with flow cytometry. And so what happens in cells is that CFTR is translated and it's degraded. And it goes through this nice cycle and it's kind of at a steady state. And the cells are glowing green when this happens. But when we inhibit synthesis with this drug called emetine, we no longer synthesize CFTR and we get degradation, and the cells have a dimmer fluorescence. And so the key experimental methods I use in lab is this thing called an emetine chase, and this enables me to quantify the percentage of CFTR remaining over time. And this enables me to look at things like half-life, which in the case of F508 delta is 40 minutes. So you stop synthesis and 40 minutes later, half of your CFTR molecules are gone. And so here's some more real world data, also from Felicia. And this is where we have F508 delta degrading nice and quickly. And this is much faster than wild type. So here we see with the wild type protein, there's a bit of a fast phase of degradation. That's because not all wild type folds correctly. But then you have this population of about like maybe 70% of wild type is nice and stable and doesn't get degraded. When we look at correctors, we see that we get something in between. So F508 delta is definitely degrading slower, but it's nowhere near the level of wild type. And then, of course, if we look at our latest generation of correctors, this vanzacaftor, tezacaftor, we get even more rescue, but again, still not as stable as wild type. And so in general, this is less stable than wild type. It's still being degraded by the cell more rapidly. And of course, my question is, what is this machinery? So to get at this, we conducted a turnover screen with CRISPR-Cas9 lockout library. So we blocked synthesis with emetine, so that enables a lot of degradation. And what we're asking is which cells glowed greener after we did this. And this is a confetti plot, at least that's what my mom calls them. And so what I hope to your take home is, because this genomics data can be very scary, is that there are a lot of processes that are coming in and degrading CFTR. We've got processes involved in the ubiquitin proteasome system, in ERAD, in the endolysosomal system, trafficking. And one thing that's kind of exciting is about 40 genes. I don't really understand what the relationship is to CFTR. So there's about 200 of these. And so they encompass all these different processes in the ER, Golgi, endolysosomal plasma membrane. One thing that was very encouraging is that we observed RFFL, which was previously characterized by Okaneda and Lukács as an endolysosomal E3 ligase. And so this is, the endolysosomal system is what's directing CFTR to the lysosome to be degraded. And this is an E3 ligase that sticks on the eat me signal. And so what happens is we knock out this gene, and we see without correctors, there's no impact. But if we compare, bright red with maroon red, we see that there's definitely slower degradation kinetics. But it's slower, but it's still not the whole story. Because if it's the whole story, it'd be like a straight line, right? But we also see a very modest increase in band C levels with this RFFL knockout. We also got some things that we had never thought to link to CFTR biology before. One of them was the endolysosomal E3 ligase MyLip, which targets the LDL receptor, which is what uptakes cholesterol into your cell. And we get something very similar where there's no response or no change in CFTR degradation kinetics when we don't knock out or when we don't add the correctors, but when we add the correctors, we do get this nice slowdown. And we also see some more band C. Okay, so what are the other modulators? Well, MyLip and RFFL are just 2 dots on this thing. So the goal one for the rest of my time in Ron's lab is trying to understand the mechanisms of other hits. And my goal 2 is to apply this technology to correct a refractory variants. And just briefly want to touch on this. So how do other variants compare with F508 delta? Like, is their response to correctors similar? Is the machinery that traffics and degrades them the same? So to look at this, I looked at just two variants. We were still collecting data on the other one. So I looked at N1303K and W57G, because I think these two nicely illustrate some fundamental principles about correctors. So if we look at F508 delta versus N1303K response, we see that if we add in these drugs, they have exactly the same effect on total levels of CFTR. Both increase quite a bit. However, if we look at surface display, we see the strong response with F508 delta. We get this very modest phenotype with N1303K. It's only about a 20% increase as opposed to like an 8-fold increase. And what we think is happening here is that the correctors are able to divert CFTR N1303K from ERAD, but it still can't effectively escape the ER and traffic to the plasma membrane. There's probably a little bit that leaks out, and that's why we can get some clinical response with this, but it's not nearly to the levels of F508 delta. Now let's look at W57G, which was some data done by my other undergrad, who is also here, Claire, and she looked at this mutation compared to F508 delta. And what we can see is that we don't see hardly any change when total levels of CFTR when we add in vanzacaftor, tezacaftor. And we also don't see any change with surface display. And so what's happening here is that this is like with F508 delta without any correctors. It's just being degraded through ERAD. It's not getting out to the Golgi or the plasma membrane. So this highlights two mechanisms of corrector resistance. One is that W57G just can't escape ERAD. It can't bind to, or it's just like it's already ubiquitinated and diverted before the correctors can act. And then the second thing that's happening is that with N1303K, it just can't escape the ER. Like it's binding the correctors, it's being diverted from ERAD, but it just can't exit. So moving forward, I'm still conducting screens with corrector-resistant variants right now. We're looking at a whole host of them right now. That's what Claire and Felicia are working on right now. I'm also working to develop methods for validating the hits in actual epithelial cell models. And with that, I'd like to acknowledge my laboratory, particularly Ron Kopito, who I've been working with now for 8 years, since I worked with him during grad school too. I'd also like to thank all of these labs on campus at Stanford, UC Berkeley, particularly the Stanford Shared Facts Facility, where I basically live in order to conduct these screens. I'd also like to thank Porteous Mila and Schlebach Labs for really giving me a lot of tools and helpful suggestions with things. I'd also like to thank the software program BioRender for helping me with my graphics. It's a huge lifesaver. And then all of my funding sources, particularly CFRI. I was very privileged in that I was actually awarded the Elizabeth Nash Fellowship 2 times. I had to reject it the first time, but it's been a great community to belong to, and thank you guys so much.